Okay, so the panel is now available for answering any questions or discussion. Do we have any questions to kick it off? Everything was crystal clear. We know everything. So I have a question that out of the series of questions that I've come up with for this particular panel. And one is, what are our limits of sensitivity? And how do we push that, especially for things like in situ measurements or even sample return? So in sample return and laboratory analysis, one thing that I think astrochemists really want to know is when we're detecting, isotope fractionation or enrichments in biomolecules, so things like amino acids or nucleic acids, whatever, where that enrichment falls in the molecule. Is it on, for example, an amine group, or is it on a carboxylic group? And can we decipher that yet?
Yeah, there's actually some work going on right now looking at position-specific isotope analysis. So I've been involved with John Eiler's group out at Caltech that's looking at, we extracted some Murchison meteorite, and he was looking at some of the amino acids within that, looking at is the isotopic enrichment in the carboxylic acid carbon versus the carbon that holds the amine group or the others. So that's a developing new technique right now, but it is something that we really are interested in because we can make these predictions about the origins, what the precursor molecules were, because we know from astronomical measurements which precursor molecules in the interstellar medium might be more enriched versus less enriched. And so then we can suggest, oh, maybe the amino acids are coming from something like Strecker synthesis versus other methods. We can predict which molecules should bear that enrichment. But we're just getting to the point now of being able to make those position-specific isotopic analyses. And I think that is going to be very interesting. But that would be a laboratory technique. In situ, I think that's going to be a long time before we can get to anything like that. I don't know if anyone else wants to.
Oh, okay. That's better. Just to follow up, I guess on the laboratory aspect, 20 years ago when I started doing amino acid analyses, chemists like to talk in moles for detection limits and sensitivities, but we were something like pushing femtomole, for amino acid. Now we've got like 3 orders of magnitude better in the last 20 years, so we're like attomole kind of limits, and that certainly makes a big difference, and I don't see why that trend won't continue 20 years from now. Thus, again, the importance of having samples available that you archive, that you keep for decades, so that when that new technique comes, that site-specific capability, we have it ready to go.
Right. But also, Jamie, it is sample limited, right? Because you need so much to be able to detect how much carbon enrichment there is, right?
Yeah. Yeah, we are sample limited. And, one of the trade-offs I talked about in my talk is sometimes we have to give up our precision to increase our sensitivity, and that can in some ways limit the questions that we can answer and how specific we can be. Things are definitely always improving. So, again, we hope that in the future we'll be able to increase our sensitivity, really get to some of these questions, but it's an ongoing struggle. That is one of the challenges.
If I may here. Also, there's a distinction here between a mass-sensitive technique, which requires a certain amount of sample in order to get the detection, and a concentration-sensitive technique that, like with laser-induced fluorescence, you can sometimes get down to single-molecule type detections. Of course, those are for macromolecules, but as those techniques progress and become more mature, they could become useful in our arsenal for space exploration.
You need a mic, yeah. You have to go up. I was told to encourage people to use microphones.
I'm really loud. For the comet flyby, or Kuiper Belt object flyby, how close will we get, and how similar will it be in terms of like images we've had of other small solar system objects, just as a comparison?
That's an excellent question, and I actually don't have numbers off the top of my head. If anybody else in this room does, please feel free to shout them out. I think it's No, I'd rather not say something that is incorrect. That's a great question, Jonathan. Thank you for that. I've sort of not been working on Pluto since the photochemical work that I've done 2 years ago, so I haven't been up to date with the latest team meetings actually. So, yeah.
It is a small object, though. It's a lot smaller than anything. What, 20 kilometers? Yeah, I think they think it's about 20 kilometers in diameter. So it is rather small. So, are there any other questions? Yeah, go ahead.
This is a question, I guess, largely for Jamie. So you kind of went through it briefly, but can you tell us a little bit about the special considerations need to be taken for archival and storage of samples to maintain their native environment, make sure there's not things like Earth-based oxidation and other things happening, as well as what are some technology gaps in the ability to archive new samples that may come into the future?
Sure, I'll start, and Danny's also done some work on this if you want to jump in. Right now, samples are curated usually in a clean cabinet with flowing nitrogen to try and keep out oxidation and moisture, and that's been pretty good. There's questions about the ability to do cold curation, particularly if we do comet surface sample return. Could we do a clean cold curation for long term? One of the interesting ways that we might be able to answer that is that NASA just announced a program this summer to look at some of the Apollo samples that had been curated in special ways that hadn't been opened since they came back in the 70s. And so proposals were due in August, and so in the next few years, they're going to be opening some of these, and some of the questions are science questions, and some are curation questions to understand if some of these samples that were stored under sealed vacuum, or stored frozen may show different amounts of contamination or preservation than the standard nitrogen purged cabinet. So I think it's an ongoing discussion. One of the things that has been very valuable with Stardust, and then OSIRIS-REx has carried this through, and future sample return missions as well, is having more and more of the witness materials so that you understand what is being contributed from the environment. You can't control all contamination, but if you have witness materials, you can understand it. And with OSIRIS-REx as well, there was a heavy contamination knowledge effort as they were building the spacecraft, of curating and archiving materials from all of the different construction phases, all of the building of the spacecraft, so that even if they aren't fully analyzed and known now, if future analyses show something, you could be able to trace it back. There is, I don't know if you want to talk about the Stardust.
I was just going to mention that, but
no, go ahead. You were doing that one, but an example where being able to go back and look at material is helpful.
Yeah, so just to address, so you mentioned contamination being of a concern, which it always is, but there's also just losing the native volatiles from the sample. So Jamie was talking about Stardust and our analyses of glycine on the foils. Well, we saw this trend that over time in curation at ambient temperature under nitrogen, we were losing glycine. Glycine is not that volatile, but what we think is happening is we were losing a volatile precursor like formaldehyde from those foils. And that could have been instantly solved by just hermetically sealing the foils. But that was never in the Stardust curation plan. And by the time we discovered this, it was sort of too late to make changes. But future missions like OREX, and if we do a comet sample return, these samples will be hermetically sealed to avoid volatile loss from the sample itself.
Okay, so I just can't stop thinking about contamination. And through everyone's talks, I'm just thinking how contamination could affect getting sampling, right? And you've talked a lot about materials, but then I was thinking about this very serene atmosphere that you painted this picture of on Pluto, right? And could we even be disturbing or contaminating that atmosphere by flying by, so creating, I don't know how close we got, right? But in future missions, maybe with turbulence or, something like that, could we be altering these environments just by getting close to them? And how does that affect the study? How do you get good information if your mere presence alters what you're trying to see?
So for the case of Pluto, I do know the details for that flyby. We got about 10 or 12,000 kilometers from its surface. It's very, very far away. And its atmosphere, Pluto's atmosphere is actually quite distended because it's a low gravity object. It extends more than 1,000 kilometers from its surface. But that's still, it's super far away. So I doubt that we contaminated Pluto at all or changed the measurement in any way.
I mean, but in the future, I mean, come on.
Oh, right.
We're
So
gonna
if we were to
back,
go
right?
orbit,
We have to at some
Team
point.
Pluto orbiter, yeah, yeah. So that is a good question, and we'd love to have in situ measurements of these aerosols in Pluto's atmosphere, and I would say that the lessons that we learned from Cassini and the types of things that we had there with the Huygens probe going through Titan's atmosphere should help inform us about the dangers of contamination. But I don't think that was a particularly large issue for Huygens. So, yeah. But Pluto's atmosphere is much thinner, so a smaller amount of contaminant may go a longer way to disturbing the data. But my gut feeling is that it won't really matter that much. Yeah.
So planetary protection is a big thing, especially in newer missions. So I'll let our panel actually talk about it as opposed to myself. But what are your thoughts on planetary protection? So this is the opposite contamination. So as you're talking about, so instead of bringing back and contaminating our samples, how do we keep from contaminating the environment? So.
Yeah, I mean, just to talk about Mars specifically, we've got to treat those samples as potentially hazardous, no matter what, when we bring them back, because we don't really know. There could have been life in these samples at one point, maybe still today. That's not really my biggest concern, just to be honest. Being infected by the Andromeda strain. I mean, the Earth itself has been hit by Mars meteorites since the beginning of time. And so we're, and some have actually argued that we might all be Martian, actually. So I'm much more concerned about what you brought up, and that's contaminating the native environment, contaminating the Mars samples that we pick up to bring back with our own terrestrial imprint. I think for me, that side of planetary protection is something that we should, and NASA is taking very seriously in the Mars 2020 rover. And I was talking to someone earlier about the fact that we have a drillable blank actually on the rover. So it's a fused silica brick that we will actually drill into and collect a core, pristine core, to understand any contamination that we added to the sample via the drilling process. So that's just one example of a control that will be very, very valuable when those samples come back. Because the bottom line is we're never going to avoid contamination. I mean, just the fact that we build a rover means there's going to be contaminants on that, and we can't avoid that. So the best we can do, as Jamie pointed out, is fly as many controls as we can to be able to subtract out that imprint.
I just want to comment. The other panelists have been focused on robotic missions. Once you start talking about sending humans to pick up samples, the contamination control becomes such a much larger concern. And I didn't show any photos of the efforts that we go through to not contaminate our samples in the field with our own anthropogenic contamination. It's one of the reasons why we go to these extremely remote sites in Iceland, is to remove ourselves from that. I think we're going to have a big problem with that once we have manned exploration of Mars.
When we've also crashed spacecraft, right, into other bodies now. I mean, comets. There's dead stuff on Mars.
Point out, I mean, Jamie can talk about all the Apollo analyses. We put humans on the moon and they collected samples, and there's clearly a biological imprint on those samples, but it didn't prevent us from finding extraterrestrial amino acids as well. So, yes, it's something that we're going to have to deal with, but we've got fancy instruments and tools and creative scientists to try to figure it out.
Yeah, really, we can never avoid contamination, but just doing as much as we can to understand it and to have ways to really measure what that contribution was of contamination. That's going to be really important. And like in some cases, we've been lucky. When I showed you the stardust with the big peak of eco, which was from nylon, and also that same nylon bags were used for many years in the ANSMET Antarctic collection as well. And so we see the same peak in a lot of the meteorites. We're pretty lucky there because that nylon only gives off one big peak, as opposed to something like latex, which would provide a whole bunch of amino acids. So understanding and minimizing that contamination background, even if you can't eliminate it completely, is going to be really important.
Oh, there it goes. So this is a very general question about this discussion of in situ versus sample return type missions. And I just wanted to hear what your thoughts were on the prospects for improving in situ measurements to the level of what we can do when you actually have a sample in the lab, given these difficulties of contamination, retrieval, the difficulty of bringing something into, onto Earth itself. A lot of the gravitational force can disrupt your sample in many ways. What are your thoughts on that?
So I don't know if you looked at my profile, but half of my lab is instrument development for in situ missions, so I have a very strong bias here. There is an awful lot of science that can be done with in situ systems with a number of different samples, especially if you have something that does not have much consumables or any consumables as your analytical tool. And here I have to make a plug for microfluidics, which does allow you to get several analyses with the same volume of reagent. As tools get better, we'll be able to look at more samples, and given my talk, you can tell I have a strong bias for more samples as well than we could ever get with a single sample return mission. However, the depth of data we could get from those limited samples with Mars sample return makes it one of these. We need both. We need more in situ missions and we need sample return would be my personal argument, and I will let these guys now argue for sample return.
No, I think you're right. You definitely need both. When the sample comes back, one of the nice things is how many different labs and techniques you can subject it to that you just can't fit on a spacecraft when you're building a mission. There's always those trade-offs of weight and power and space and all of that. But there's so much that you can learn from the in situ measurements. You need to have a mix of those with the sample return. I think sample return is great because you can get all of these measurements, because you can have these samples stored for so many years. The Apollo samples that I looked at for the amino acids, I wasn't born when those were collected, and we used instruments that didn't exist at the time. So sample return is important, but we are making really good progress in miniaturizing these instruments and being able to do a lot of very good in situ measurements as well. I think you need a balanced portfolio.
There's also destructive techniques that we have for our samples. So what are your thoughts on we're destroying things that we're actually analyzing?
Yeah.
Yeah, the joke with Viking and Curiosity was, we sent these rovers to Mars to look for life, and then they killed it, by heating the sample and obliterating it. There certainly are techniques, Raman spectroscopy, non-destructive techniques where you can get at some of this information. Ultimately, if we're really looking for the complex biosignatures, getting at polymers and things more like DNA, we're going to, at least right now, we got a result to some sort of wet chemistry extraction approaches, which aren't completely indestructive. You're not obliterating the sample like you would by heating it in an oven, but even if you expose these samples to solvents, that's considered destructive analysis by NASA.
So I think there's probably a lot that could be said about instrument miniaturization. And just hearing about what SAM can achieve in just something the size of a microwave, that's interesting. But I'm going to take it a little bit of a different direction. So, Amanda, you may have hit it talking about microfluidics, but I was actually thinking what we could learn from what industrially we do with high throughput chemistry, right? All these small volume, 150 well microtiter plates. What can we learn from that as far as taking that or something even better like microfluidics and tailoring it to some application to measure in situ?
Okay, so now you've set me up for a shameless plug for one of my favorite instrument systems that is currently under Matisse development to get to TRL6. It is the Enceladus Organic Analyzer being designed out of Berkeley Space Science Lab that incorporates a microfluidic sample handling processing system that's capable of addressing multiple different samples for multiple different compound classes, specifically using a microfluidic approach and capillary electrophoresis with laser-induced fluorescence detection, which was the technique responsible for us solving the Human Genome Project under budget and ahead of schedule. So in terms of miniaturizing these sorts of things, the overall box is about this big, and we're really looking forward to getting some really good data out of that as we elevate the TRL. And of course, TRL6 is not mass spec, which is being used primarily in these Mars landers up here. Keep an eye out. Can I make another shameless plug while I've got the mic? Down here at the very low, are you familiar with TRL, by the way? Okay, TRL technology readiness level, it goes from about 1, where you can write it on a napkin, up to 10. We've flown it in space and it has successfully worked multiple times. TRL 6 is a level where we have demonstrated it in a relevant environment or multiple relevant environments for the eventual launch here on Earth in spacecraft-based laboratories. You go from 6 to 10 via successive different demonstrations in space environments. So that relatively high TRL instrument, the EOA that I'm talking about, we're trying to go from about 4 to 6 by the end of that development. Most of my research group focuses on those crazy ideas from the 1 to 3 level, and we've just got some really good data showing that we can build a miniaturized, just laser-induced fluorescence detection system in a little 500-gram capsule that's about 4 inches across by about that tall, that is capable of surviving a 50,000 G impact that's 5 kilometers per second with a 50 millisecond stop time against cryo ice. This is about the hardness of granite. And so this would be good for the icy moons and Pluto and other places like that. It looks like we can build it and make it survive. We've tested up to 50,000 Gs.
Just real quick to follow on to Amanda's comment, this instrument she's talking about, microcapillary electrophoresis with laser-induced fluorescence detection, extremely, extremely sensitive. This is what I was talking about, getting at the atomal. So you mentioned the SAM instrument, great tool, right? We might be able to detect amino acids if they're there with this wet chemistry, but we're, again, at the picomole, kind of detection limit, and orders of magnitude higher sensitivity. So, yeah, wish you luck getting this thing ready for flight, because I'd love to see it go to Mars, Enceladus, elsewhere.
Are there other questions? Yeah, go ahead.
So what chemistry work in the lab needs to be done to prepare for these missions? I mean, so I guess it's sort of trying to find the common ground between the chemists. Is there research areas that are really missing for these in chemistry that we should be trying to focus on? But in our labs on the ground, what's the chemistry that's missing that you need for these things? You're just going to get whatever you can get and hope that the chemistry works out when you get there? Or...
In terms of the ocean worlds and the icy moons, I think that our understanding of the catalytic properties of metal-bearing minerals is still pretty low, and I think that in the lab, through techniques of trying to simulate hydrothermal environments, that's very exciting to try to understand what the catalytic properties are of these and their role in possibly creating organics, definitely creating organics, but the organics that are also relevant and specific to life. Something that we should work on.
Are there other questions? So we do have a limitation in planetary science, is we get one big mission every so often, or we can only go to one target, or so this becomes a resource issue. We also have other, not only funding resources, but also power. If we want to go to the outer solar system, solar panels are inefficient. So we're looking at how to get there. Communications. Also, we're in an era of big data. We're collecting all kinds of data now, and getting that data back on Earth for analysis is something that's really a challenge. So just curious what our panel's thoughts are on these kinds of things. Give us money. All right, that's the answer. Give us money.
I mean, I've been at NASA for the last 2 decades, and it's just amazing what we're doing right now. I know, the public, they tend to get sidetracked if we're not flying humans into space and what's NASA doing, but you look at all the robotic missions, Pluto for the first time, organics on Mars, exploring asteroids, the outer planets. I mean, this is really the golden era of robotic exploration, and what we're learning about the solar system and now exoplanets, and hopefully JWST will provide more insight there. It's a really fun time to be in planetary exploration.
But the prioritization of missions, is another challenge, right?
Okay, so I think you hit on something very important that chemists can totally help with here, which are a number of the different limitations of the material and power technologies that we're looking at when we go to these extreme environments. We need batteries that can survive at much colder temperatures than they currently can, and that perhaps can provide power over a longer time frame. Materials as well for these extremely cold environments or the extremely hot environments. We didn't look at Venus at all here, but that's an incredibly challenging environment for in situ science. And as a part scientist, part engineer, those material limitations are things that I face against relatively regularly.
I think a general note, too, is to expand and improve on what's called the Deep Space Network, or DSN, which is a set of antennae that are communicating with all of our spacecraft. And they're kind of aging right now. And it was a panel at the Division for Planetary Sciences meeting just last month that noted that the younger generation, the kids these days who are going to grow up and become scientists, what they want is not just pretty pictures, but video. That's what they expect out of spacecraft. And so in order to get not just high definition pictures, but like video coming from the next Europa mission or Jupiter mission or whatever it is, we would need to expand our capabilities to communicate and downlink. And just personally working on Pluto, it took something like 18 months to get all of the data back from that farthest region of the solar system to Earth. And so that's just kind of the time frame that we're working with the technology that we have right now.
And just to follow up, this is an area where something like laser comm and that technology may help get a lot more data back.
I work on instrument development too, so I just wanted to emphasize a little bit more what Amanda said. We suffer from something called the tyranny of the rocket equation. So the heavier something is, the more fuel we need to get it off of Earth, but then the rocket is heavier, and so eventually you lose, and you can't get off of Earth, which is bad because you need to get off of Earth to go to Mars, for example. So things like radiation-hardened hardware are really important because then that minimizes how much shielding we actually have to send on the spacecraft, which is heavy and isn't doing anything besides sitting there shielding. Things like cryogenic valves or valves that can operate at high temperatures. So like anything you can imagine that could be made lower mass, rad hard, operate under high pressure, low pressure, high temperature, low temperature. We're just always struggling against those kinds of things because most of the technology that we use in our lab instruments wasn't designed for those kinds of things. It wasn't designed to be radiation hard or anything like that. And so it's not just applying the technique in space, but we have to figure out how to actually fly an instrument that can survive those environments, that can physically get off of Earth because it doesn't weigh too much. So I think those are also things to consider, especially for like Europa exploration. We really need radiation hardened hardware to help us with that. And so that's something else that is important for us.
Any other questions, comments? Go ahead. Yep.
And one more thing, I think a general theme that we've seen here is there's an awful lot of data, and there's whole groups of data science chemists that know how to handle these massive amounts of data and perhaps mechanisms for improving the interaction between the two groups could benefit both sides.
So, given the subtitle of our workshop, I'd like to hear from each of you. So, from your discoveries, how would you say those have informed the chemical sciences challenges that we face today on Earth?
I may have just stated mine, where we have these very large data sets that we've been trained as chemists to try to interpret them visibly or just applying a linear regression to them. But the actual lessons that we're trying to get out of them are much bigger than that. And all of these data reduction techniques and training of tools to tell you predictively, is it this or is it that? And with what percentage can it assign it to those classes? I think within my own maybe narrow field of research, that seems to be incredibly important for spaceflight in identifying exactly what are biosignatures. How would we look at a set of data and determine, yes, this is a biosignature or not? And also understanding what is habitability and how do we define that sort of a thing. I'm going to stop now.
I guess I have a thought. I'm not sure if this exactly gets at your question, but just origin of life in general. Like, how did we get here on Earth, right? How did life start? One of the problems we faced as organic chemists is that the oldest rocks have been heavily processed through plate tectonics and metamorphism, and so that record the prebiotic record has been pretty much destroyed here. But on Mars, we've got an environment where you didn't have substantial plate tectonic activity. We've got 4 billion-year-old mudstones, a record that's been pretty much erased on Earth. And so we could potentially learn about the original life on Earth, on Mars, strange enough, or Titan, for that matter. I mean, prebiotic chemical factory there, so we need to explore Titan as well. So anyway, I think just learning about our own origins, and because that record has been destroyed here, we kind of need to go elsewhere to look for it.
Let's talk about the samples that we look at, which are these really complex mixtures of organics that really, very diverse and in very low abundances. I don't know if that's answering your question at all, but it's a challenge that we are facing, that we can use chemical tools that are being developed in a lot of different fields on Earth to try and figure out how do you look at these complex mixtures that are present in very trace levels. One of the things that bothers me most about our research is we don't have good terrestrial analogs. Like, we'll spend 2 years developing a method using standards or using our best ideas, but then we get to an actual sample and there's something totally unexpected there that kills our method or doesn't work. And so this idea of how do you analyze this very complex mixture that is not only low abundance of the molecules you're interested in them, but low abundance of the sample itself, so that you don't have a lot to play around with to develop, is an area of challenge that I look to the chemical sciences community in general for ideas on how to help.
Sure, I feel like this is just a repeat of my my takeaway messages, but I feel like Pluto is giving us insights into organic chemistry in a brand new pressure temperature space. And with the icy moons, I think we're trying to understand whether geochemistry, as we understand it here on Earth, is common across wet, rocky worlds in the solar system, and perhaps extended beyond. And I think we're finding out that yes, the processes here, such as serpentinization, will happen on other wet terrestrial bodies. And the next question is, do those processes lead to life?
And I'll just say, as an astrochemist in general, it's the unknown unknowns. Like everybody has said over the last few days, when you give, the opportunity, chemistry can do anything it wants in any of these environments. And it's often the things that we don't expect, which end up leading to the best discoveries or the new insight and things that we don't know. So how you plan for that and how you anticipate that is a challenge. How do we build a telescope to detect things that we don't know that exist? We get lucky sometimes that, you know, things fall into place and we can actually see them, but that's not often the case, especially for in situ, or as Danny was explaining with, you know, the landers is, you know, if we're not prepared for a certain kind of chemistry, then we don't have something there to detect it. So it's, you know, how do you broaden that scope? Yeah.
So this is meant to just sort of be a fun question. What do you think would be the most interesting astrochemical surprise to discover that would not involve extraterrestrial life?
I think non-organic chemistry. And so something like extreme inorganic chemistry, I think that would be a really cool discovery. No thoughts?
Well, I guess to flip it around, I think not detecting some sort of biology in our solar system would be surprising to me.
Yeah.
Sorry, I've got a general question in the vein of what Jennifer had said. So, you know, we've talked a little bit in the breakouts about, you know, challenges getting funding related to astrochemistry, as well as differences between the U.S. academic system versus European in the field of astrochemistry. So you've got a room full of undergraduates who are uncommitted that are looking to go into grad school. What advice could you give them to inspire them to pursue a career or a graduate education in the field of astrochemistry? What's in it for them?
Free trips to Iceland.
Mic drop. And Hawaii. And Chile, right?
I mean, I just love the questions that we're trying to answer. It's just scientific, you know, it's satisfaction in trying. I mean, a lot of my work is this astrobiology, trying to figure out the origin of life. And it can be a challenging funding environment, and there's a lot of challenges involved, but it's these questions that people have asked and wanted to figure out for a long time. I tell people what I do, you know, almost everybody just is very interested in it, is really excited about it. And so I can't give the students necessarily a really concrete, you know, personal gain reason why they might want to go into astrochemistry, but I think that these questions that we're trying to answer are really big questions that are exciting.
I mean, just to follow up on that, I mean, we haven't found definitive evidence for extraterrestrial life yet, right? So they could be part of that team that eventually does that. If that can't excite them, I give up.
But people do come into this field out of a deep-seated love for the topic. I mean, these are the biggest questions that humans have been asking since we became human. Are we alone in the universe? What is the origin and evolution of life? And, you know, if we find the answer to are we alone, and it is an independent emergence of life, that would be a second data point, because currently all life on Earth is evolved from a single common universal ancestor. That would help inform us with a universal theory for what is life and what makes up those characteristics. And these are just such amazing questions. And as a person advising students, it's kind of find the thing you find deep meaning in doing and the thing that you enjoy doing. See if you can combine those things and then convince someone to pay you to do it. And that's like the secret to happiness, right? Anyway.
Well, I definitely agree with all of the things that have been said. I guess from a personal perspective, what inspired me to go into the field of planetary science. I took my first planetary science class in the spring of 2009, which was the spring that the Kepler spacecraft launched. And I remember my professor showing a video of that launch to our class, and that was a really moving experience to me. I was really drawn not really to the rocket that was going up into space, but seeing all of the scientists and engineers, the people at NASA who poured their hearts into the spacecraft, cheering and just in tears as they were watching this thing go up. And I was moved emotionally by that, and I really was like, I just wanted to be part of that community who felt so passionately about space exploration and built a modern wonder, technological wonder, and to be able to get to use modern technological wonders, data from the Cassini spacecraft, data from New Horizons, is just astonishing to me. And every once in a while, I have to pinch myself and be like, wow. What am I doing? I can't believe I'm doing that. so yeah, showing people that aspect of space exploration too, getting to use the awesome technology, is really motivational to me.
So this is not about, it's not a workshop on the origin of life, but it drives what some of you guys are doing. And so let's fast forward, and we have concrete evidence for bacterial life, for example, on a place outside of Earth. What changes for us in terms of understanding where we're coming from and answering that question? To me, it's still not answered. It just says there's a microbe on XYZ and it has a different metabolism and it's not DNA based, but it has something else. It doesn't use ATP, blah, blah, blah. What has changed for me?
Okay. May I just ask, are you familiar with any of the work out of the Center for Chemical Evolution at Georgia Tech, or Dave Deemer's lab, or the Mike Russell group at JPL? Okay. So let's say that we found life at one of these icy moons where there are no warm, wet ponds or surface hydrothermal activity. That would start to place some constraints on the relative importance of the contribution of those environments to the chemistries that are involved in the emergence of life. If we found life on Mars and it didn't have DNA, but it had something else, now that's an environment where these warm, wet ponds and surface hydrothermal activity come into play, and it could minimize the amount of these deep submarine alkaline hydrothermal vents in the emergence of life. Additionally, the different chemistries that have been looked at by these emergence of life groups all recognize that the current DNA, RNA, protein system is very, very likely an evolved characteristic. And so there must have been something before RNA, before DNA, and before proteins. And what were these protopolypeptides? What were these protopolysaccharides? And all of these different things. If we could find a different emergence of life that had different of those peptides, and polymers, then we might be able to backtrack what were those original structures prior to the evolution of the current polymer environment with these three main polymers.
Oh, go ahead.
Oh, just real quick, I think that what it would do for me is help strengthen my bond to the rest of humanity and the rest of life on Earth. I once wrote a poem that I can't remember all of it off the top of my head, but one of the lines was, if we discovered alien life out there, maybe we would all realize that we all shared the trait that we all have 5 fingers because maybe that alien only has 4. You know, that we start realizing the commonalities between us even more viscerally, and start to break down these invisible walls that we've put up between ourselves. And maybe we're just going to find microbial life out there, and that's fine, but the analogy between 5 fingers and 4 would be maybe they use a different set of amino acids and look at all of life on Earth using this common ground of biochemistry that connects me not just to you, but to the plants outside and to the bacteria and archaea in my gut, just to realize that we're all very united as a story of the evolution of the universe and of our planet, our spaceship Earth.
I guess just to follow up, I don't know if you're familiar with the Drake equation, but it's basically a way of predicting what the probability is of having an intelligent life somewhere in the universe. And one of the weak links with that equation is the probability that you can go from a non-biological chemical system to a biological system. This is something that, at least not yet, we haven't been able to do in a test tube, to my knowledge. If anybody is aware of that, let me know. That'd be pretty cool. But if we did find a bacteria on Mars or Europa or Enceladus, and we really were able to show chemically that it was an independent origin, okay, it wasn't life on an Earth rock that seeded Europa or whatever, that it really evolved independently, man, that would say, okay, it's happened twice now in one solar system. And I would say the probability just went way, way up for extraterrestrials somewhere out there. So I don't know if that would impact it. Certainly, look, finding a microbe on Mars would not be the same impact as an alien visiting us, right? An intelligent life. I mean, let's be real. But if we could show it's an independent origin from an original life perspective and just probabilities of is there life elsewhere, that'd be a huge discovery.
And I think the implications for how we search then for life outside of the solar system would be a huge impact, right? Because then we would know that there's potentially different environments. So now we're not looking for the Goldilocks zone in exoplanets anymore. Now we know that we can actually look for things like habitable signatures or biosignatures in different types of environments. And I think that would really expand what we're trying to do with future missions. Because right now, everything we're designing, we're trying to find water. Or a surface with, an ocean and an atmosphere that's at a certain temperature so we can have liquid water. If we find that there's life like in gas giant atmospheres, then wow, look at the whole new realm that we can actually start looking for things that are, as you were saying, the atypical behaviors of the chemistry in atmospheres that would be true indicators of something going on, potentially life.
And one more thing, we keep saying independent origin, but even if it is of a similar ancestry to us, but not direct contamination, that would indicate that there were mechanisms for biology to move between planets that were not us bringing it with us. And so that's a meteoritic carrying of material around that's panspermia. That would be a very interesting result in itself as well. It doesn't answer the how does life begin and emerge question. It simply says the environments we have on Earth are possibly not the environments that lead to the emergence of life because we could have gotten here from somewhere else.
I think we should wrap up our panel though. So thanks to you all.